Microbiology, laboratory diagnosis. First we do microscopic test. The first one is peripheral blood smear. Peripheral blood smear examination is a gold standard confirmatory test for detection of malarial parasite. Peripheral blood is collected from ear lobe or by finger prick in older children and adults and from the great toe in infants. Blood should be collected few hours after the height of paroxysm of fever and before taking antimalarial drug. Types of peripheral blood smear: thin and thick smear. Stain: they are stained by one of the Romanowski stains such as Leishman-Gainsa and Field Wrights or JSB stain. Examination: both the smears are examined. The thin smear is screened near the feathery tail end. At least 200 to 300 oil immersion fields should be examined before the smear are considered as negative. Advantages: peripheral smear is rapid, simple and cheap. A thick smear is useful in detecting the parasite, quantification of parasitemia, demonstrating the malarial pigments. A thin smear useful in the speciation of malarial parasite. Speciation of malarial parasite: the speciation by thin smear is based on the detection of ring forms, schizons, gametocytes, type of pigment produced and RBC size. Ring forms help in accurate speciation. It comprises a vacuole in the center, a peripheral thin rim of blue cytoplasm surrounding the red nucleus. Plasmodium vivax ring occupy 1/3 of RBC size. Late trophocytes are amoeba shaped. In Plasmodium, oval ring form same as Plasmodium vivax but in oval RBCs. In Plasmodium, malaria difference is in late trophocytes, which is a band shape. Plasmodium falciparum rings are smaller than in Plasmodium vivax. Three variant of ring form are seen: multiple ring form inside the same RBC, acyl ring form attached to RBC membrane, double dot headphone shaped ring form. Schizoids are not seen in peripheral smear. Gametocytes is crescentic or banana shaped and larger than RBCs, whereas for other species it's spherical and occupies almost the RBCs. Buffy coat examination: blood is collected in capillary tube, then it is centrifuged and examined at the buffy coat region under UV light. Parasitized RBCs appears as brilliant green dots. WBCs also taken up the stain. However, normal RBCs don't taken up the stain. Faster and more sensitive but expensive. Rapid diagnostic test based on the principle of ICT test. Antigens: malaria RDT kits are designed to detect several AG of Plasmodium species. LBH and Dedolus. This AG are produced by all Plasmodium species. However, newer kits can differentiate between the PanelDH and PFLDH. HRP2 histidine-rich protein only produced by Plasmodium falciparum. Principle: these kit contain 2 parasite detection lines and 1 control line. Test line 1 coated with capture up specific for Plasmodium falciparum. Test line 2 for all Plasmodium species, example PanelDH or aldolase. Culture: the culture techniques are mainly used for the preparation of malaria antigens, not for molecular methods. Various molecular tests has been used. 1. Nested multiplex PCR. 2. PCR can also be used to detect drug-resistant genes. 3. Real-time PCR is useful for quantification.